My name is Neha hambire. My topic is history of plant tissue culture. History of plant tissue culture. Historical Henry Louis Duhamel du Monceau pioneered the experiments on healing in plants through spontaneous callus formation on decorated region of plant. Next point, after propounding the cell theory by Skelton and Scaven, suggested the presence of polarity, key feature that guide the development of plant fragments. He observed that the upper portion of a piece of stem always produced buds and the basal region produced callus roots. In 1902, a German botanist, Gottlieb Herbarer in Berlin, developed the concept of culture of isolated cells of Tradescanta in artificial conditions. Next point is laboratory condition requirement. First point, washing and storage facility. Large sink with provisions for hot and cold running water. Distillation apparatus, washing pipette, washer, dryer, and cleaning brushes, etc. Media preparation room, space for keeping chemicals, labware, culture, vessels, closure, and miscellaneous equipment requirement required for media preparation and dispensing. Transfer area consisted of UV tube, UV light from the tube and use of 95% ethanol helped to maintain sterile condition inside the space of plastic box. Maintenance of aseptic environments. Following are some sterilization methods for aseptic manipulation of plant tissue. First, sterilization of glasswares like petri plates, flask, pipettes, etc. and metallic instruments are sterilized in a hot air oven at 160 to 180 Celsius for 2 to 4 hours. Next, sterilization of instruments. Metallic instruments are flame sterilized, that is, dipping into 75% ethanol followed by flaming and cooling. It is called incineration. Sterilization of culture room and transfer area. Washed with detergents, the 2% sodium hypochlorite or 95% ethanol. Larger surface area is sterilized by exposure or to UV light. Sterilization of nutrient media. Sterilized by autoclaving and 50 psi for 30 minutes. Sterilization of plant materials. Surface of all plant materials have microbial contaminants, therefore it is used to make plant material sterilized. Next point is tools and techniques for tissue culture. Following types of tools and technique used for plant tissue culture. First point is pH and pH meter, defined as negative logarithm of the hydrogen ion activity, but in practice usually hydrogen ion concentration. Next point, autoclave. First, used to sterilize medium glassware and tools. Sterilization of material is carried out by increasing moist heat 121 Celsius. Moist heat kills the microorganism. Third point, plant growth chamber. Constructed in a suitable sized room or can be purchased as commercially available equipment. Fourth point, laminar airflow bench. It is the main working table related in plant tissue culture, filled with high efficiency particulate air. Filters, HEPA filters have a very small bore size, 0 to 3 micrometer. Microscopy, electron microscopy, study of subcellular organelles. Scanning electron microscope provides surface view of whole structure of specimen. Light microscopy is absolutely indispensable tool for cell biologists. Fluorescence microscopy, selection of fluorescing secondary metabolite rich cell. 6-point colorimeter. It is used to determine the concentration of biochemical compound is colorimetry. 7-point centrifugation is an instrument which produces a centrifugal force by maintaining the samples around a central axis with the help of an electric motor. It is widely used in analytical technique preparation. Chromatography. It is the technique to separate molecules are on the basis of differences in size, shape, mass, charge, and adsorption properties. Thermometer. It is the device that measures the temperature. Next point, methods of sterilization. First point is asepsis. Maintenance maintenance of germ-free system in all the operations known as asepsis. Surface sterilization of explant. Most contamination is introduced with the explant because of inadequate sterilization or just very dirty material. In laboratory, in principle, three types of sterilization is used. First, dry heat. Second, wet heat. Third, filter sterilization. Culture media and their constituents. Vital activity of a cell is the absorption of nutrients through cell membrane and rapid proliferation into innumerable cells. White 1934 observed in the unlimited growth of isolated root tissue when provided with nutrient medium containing inorganic salts, sucrose, vitamins, growth hormones, and a few amino acids. Components of nutrient media. Inorganic minerals. It includes micronutrients in the form of salts in large amount and microelements. Growth hormones are non which stimulate the biological activity is cultured material. Organic constituents. The organic compound serves a source of carbon and energy. Vitamins are required in as they catalyze the enzyme system of the cells. Vitamin B1 is most commonly used vitamin for all plant tissue cultures. Other group of vitamin which stimulate growth are in niacin, vitamin B2, B6, CH, and B12. Amino acid most widely used are L-aspartic acid, L-asparagine, L-glutamic acid, L-arginine. Solidifying agents. Most commonly, agar is used as solidifying agent. Agar gel do not react with constituents of media and not digested by plant enzymes of media and not digested by plant enzymes generally 0.15 to 1% agar is used to form gel. pH The pH affect uptake of ions. Optimum pH between 5.0 to 6.0 is required for growth and development. Types of culture. First point, callus culture. Callus culture. Callus developed by infection of microorganism from wounds due to stimulation by indigenous growth hormones and auxins and cytokines. However, it has been artificial developed by adopting tissue culture. Explant a 2 to 5 millimeter sterile segment excised from a stem tuber or root is transferred into nutrient medium and incubated at 25 to 28 degrees Celsius. Nutrient medium supplemented with oxygen induces cell division. Soon the upper surface of the external of explant is covered by callus. Callus is an amorphous mass of loosely arranged thin-walled parenchymal cell. The unique feature of callus is that the abnormal growth has biological potential to develop normal root, shoots, and embryos, ultimately forming plant. When callus after a long has growth on nutrient medium, time it becomes essentially to subculture it within 28 days on a fresh medium. Anther culture. Second, anther culture. Anther, a male reproductive organ, is diploid in chromosome number. The aim of anther culture and pollen culture is to get haploid plants by induction of embryogenesis. Haploid plants have single complete set of chromosomes that in turn may be useful for the improvement of many crop plants. Sunderland 1979 has described that the anthers to be cultured should be one of the three categories post-meiotic, that is pre-meiotic and meiotic. Culturing technique. Anther are superficially sterilized and washed with double distilled sterile water. They are excised from the flower buds and their proper developmental stages are determined under microscope. Micropropagation. Callus culture means micropropagation. Mura sheets significantly contributed for establishing micropropagation technique and its further biotechnological application. Murasi's 1974 has described the development of micropropagation into three different developmental stages. Stage first, establishment of explant aseptically. Stage second, multiplication of propagation by repeated subculture on specific nutrient medium. Stage third, rooting and hardening of plantlets. Foster 1987 have described the following four stages of micropropagation. Stage first, selection of suitable explants and inoculation into nutrient medium. Stage second, multiplication and growth of culture, which take about 2 months followed by repeated subculture. At this stage, rootless culture are obtained. Stage third. At this stage, culture are obtained by changing the medium to planting out in the three substages. Diagrammatic representation of shoot tip in micropropagation. Hardening. The term hardening refers to any treatment that results in forming hardening of plant tissue. Such treatment reduces the growth rate, thickness, or the cuticle and wax layers, reduces the percentage of freezable coater in the plant and often results in a pink color in stem, leaf veins and petioles. Methods of hardening. Water lightly at less frequent water lightly intervals, but do not allow the plants to with severely. Expose plants to lower temperature than is reported as optimal for their growth. Biennials are exposed to cold for an extended period. They may bolt in lieu of developing properly. Do not fertilize put with nitrogen immediately before or during the hardening process. A starter solution or liquid fertilizer could however be applied to the hardener transplants 1 or 100 days prior to transplanting into the garden. At the time of transplanting, gradually expose the plants to more sunlight. This results in the more development thicker cuticle layer, thereby reducing water loss. Thank you.